First, I'd like to thank the organizing committee for allowing me to present this short paper, and just thanking them for allowing a poultry paper to slip through the cracks and make it on the program. I was just glad the presentation wasn't on Saturday. So what we're talking about today is water holding capacity. I really appreciated Dr. Warner's talk this afternoon and just how complicated it is. Well, in the poultry area, one of the things we're interested, it's been a problem for a while, is inferior water holding capacity in the breast muscle. In the U.S. Market, the breast fillet, boneless, skinless fillets represents a large portion of the value of that carcass. And unfortunately, pectoralis muscle is highly susceptible to developing poor water holding capacity, primarily because it's about 100% type 2B fast glycolytic muscle fibers that undergo a very rapid rate of post-mortem metabolism. Now, I'd contend going through the literature that the mechanisms controlling water holding capacity, specifically in chicken, aren't as well established as they are in pork or in turkey. If you look through some of the literature, most of the references that are made in the chicken papers simply reference what was done in the PSE work in the pork and in the turkey area, and I would contend that it doesn't always apply. Now, while most of the basic mechanisms do apply and there is transfer over from within the species, there seems to be some subtle differences, and partially the reason for that is the uniqueness of the chicken. Like I said, it's about 100% type 2B fibers, but then it's also the size of the breast muscle as well, that even though it undergoes a fast, rapid post-mortem metabolism and goes into rigor sooner, you can also chill it down much quicker because of the size of it. They're also slaughtered at a much physiologically younger age with not a lot of connective tissue in there. And I think one of the areas where this discrepancy comes in, and you see it in the literature, is in the area of protein denaturation and the role that it plays in the water holding capacity. It's well known in pork and turkey. Classic PSE data tells us that if we get increased protein denaturation, specifically myosin, myofibrillar proteins, we get a poor water holding capacity. Well, in a lot of the poultry papers, they reference this as well and say this is the phenomenon, but then you look at the data and it doesn't match. And some of the key studies that have looked at this point this out. In a study by Van Lack and Lane, in vitro study where they isolated myofibrils from both pork and chicken, and then they looked at them in vitro post-mortem conditions, they found that indeed the pectoralis muscles from the chicken seem to be more resistant to denaturation, specifically the myosin. And later they looked at obtained pale and normal color breast fillets. They looked at protein solubility as their measure of protein denaturation, and they didn't really see a lot of differences there. So you put these two studies together, and it seems like myosin denaturation isn't as big a factor in chicken breast muscle as it is in the red meat species. Later on, though, in 2005, a study came out by Barboot where they looked at the protein extractability in pale, normal, and dark fillets, and what they found there is they actually found the opposite, and something that would fit more with the red meat, where they saw more protein denaturation, if you will, in the pale breast muscles. In a more recent study, Zhu et al. In 2011, where they deboned early and then incubated them at different temperatures, and what they found in this was that the incubation at high temperatures early post-mortem caused glycogen phosphorylase denaturation, pale color, and lower water holding capacity in the breast fillets, and this pointed out and highlighted the potential role of the sarcoplasmic proteins in the water holding capacity. Now, a phenomenon we've been a little more interested in our lab over the last couple of years is looking at the role of post-mortem time in the poultry area. And one of the things we found in a study earlier this year, we saw that in early deboned fillets, if we look at water uptake percentage as a measure of water holding capacity, early on we see a slight increase, and then we see a decrease in the water holding capacity, probably due to the rigor mortis setting in, and then a gradual increase out to 24 hours. And we know over this time that myofibrillar protein solubility is decreasing as well. So our basic question was a very simple one. Does our relationship between water holding capacity and protein denaturation, or protein solubility as our measure of that, does that change with postmortem time? We did this in a very simplistic approach. I try to keep things simple. And our basic question was, can we determine the relationship between water holding capacity and protein solubility in broiler breast fillets of varying meat quality at different times post-mortem? So our goal in this was to get varying meat quality as far on the spectrum as we could, and then look at protein solubility. So a very simplistic approach. We did this, we wanted to keep it as in vivo as possible and not incubate it at certain temperatures to get this. So we collected boneless, skinless breast fillets from a commercial processing plant. These were taken from 5 to 6 week old broilers, smaller side 5.5 to 6 pound broilers, and we collected these after they were water chilled and deboned, and we collect them about 2 to 2.5 hours post-mortem. And I should note that this particular plant used electrical stimulation early to hasten the onset of rigor to allow the early deboning. We sorted them based on color, pale and dark, representing our high and low water holding capacity, and we did this over three replications, brought them back to the lab, maintained them at 4 degrees C, and measured color on the bone side and pH at 24 and 4 hours. We used 3 basic measures of water holding capacity. First, I should say that we designate each side as a 6 or 24-hour postmortem. We measured drip loss on a 30-gram core out of one side, starting at 6 hours, weighed the samples back at 2 and 7 days postmortem. We used a brine uptake assay as a measure of salt-induced water uptake, and then we used that swollen pellet from that as a measure of cook loss in an 80-degree water bath for 20 minutes. For this study, as a measure of protein denaturation, we used protein solubility. The standard assay that we used was sarcoplasmic protein in a neutral salt potassium phosphate buffer. We measured total protein solubility in a neutral potassium phosphate with 1.1 molar potassium iodide, and then the myofibrillar solubility we calculated from the difference. We also isolated the myofibrillar and the sarcoplasmic protein fractions, ran them out on gels, and then we were also interested specifically in the glycogen phosphorylase and how that was behaving, so we ran parallel Western blots just to verify identification. We analyzed this as a two-way ANOVA, simply looking at the water holding capacity groups and our postmortem time as our 2 factors, and we used carcass as our random effect. And quickly what we saw was no surprise here. We sorted based on color, trying to get various water holding capacities. First thing we noticed was the size difference. Just sorting on the line, the pale color fillets on average were about 25 grams heavier. No surprise, we measured color. We had 61 for the L-star value for the pale fillets and a 46 on average for the dark fillets. Our pH ranges for our dark fillets were 6.2 to around 6.2 at both 4 and 24 hours post-mortem, and our pale fillets started around 6, and at 24 hours post-mortem they had dropped to about 5.8. Our measures of water holding capacity, and through the next several slides, the blue bars represent our dark color fillets, or our high water holding capacity fillets, and the red ones are the pale. And what you can see with the brine uptake, and just a reminder, the brine uptake, the higher this percentage, the higher water holding capacity. What we saw is no surprise. We saw a higher water holding capacity in our dark fillets, about a 3-fold, both at 6 and 24 hours, and we saw an increase in water holding capacity from 6 to 24 hours. Looking at our cook loss, we saw that the pale fillets had a slightly higher cook loss at both 6 and 24 hours post-mortem, but that the post-mortem time didn't really have a strong effect. Again, no surprise, looking at the cumulative drip loss, we saw that pale fillets drip more over the 2 to 7 day period. So what we have here is 2 distinct populations of breast fillets, high water holding capacity and low water holding capacity. So then the question is, how does that affect protein solubility or protein denaturation? And the first thing we found looking at myofibrillar protein solubility, simply despite these 2 widely varying, and we would kind of argue based on what we see in the literature and the plant, about as far apart as you can get in water holding capacity, myofibrillar protein solubility is the same at 6 and 24 hours. There's a slight decrease in both the pale and the dark from the 6 to 24 hours. In the sarcoplasmic protein solubility, it was a slightly different story. The dark fillets had a slightly higher solubility at both 6 and 24 hours, and there's a slight increase in the protein solubility over this 24 hours. So how did this relate to our water holding capacity measures? We just wanted to take a simple approach, look at basic correlations. It doesn't show cause and effect, but we just wanted to see what these relationships were. And no surprise what we saw here, whether we're looking at the myofibrillar solubility related to any of our measures of water holding capacity, there's virtually no relationship, whether we measured it at 6 hours or 24 hours. The sarcoplasmic protein solubility, we saw similar relationships, the only difference being with the drip loss, we saw a slight significant correlation there. We wanted to take this a little bit deeper and look at the glycogen phosphorylase. We know in both turkey and pork that as the glycogen phosphorylase denatures, we see a tendency for it to translocate, if you will, from the sarcoplasmic to the myofibrillar fraction during isolation. And we wanted to see if this was happening in the chicken breast muscles that had been deboned and chilled in a plant setting. So we ran out the sarcoplasmic myofibrillar proteins on gels. We ran the Westerns to verify our presence, and what we could see over these 72 birds that we analyzed that we did see this phenomenon going on in the chicken. We saw this, and what we tended to see was that in the low water holding capacities, we saw more denaturation of the glycogen phosphorylase protein. Excuse me. And putting some numbers to this, we quantified based off our SDS page numbers, and what we found with in this area is that we saw a big difference between the low water holding capacity, or our pale fillets, and our dark fillets. In the sarcoplasmic fractions, when we looked at those, we saw a much higher content of the glycogen phosphorylase in the dark fillets, and the inverse was true in the myofibrillar fraction. And this didn't really matter if we were looking at the 6 or the 24-hour samples, we saw this relationship. So this basically told us two things. One, that in the pale fillets with a low water holding capacity, we were seeing a much higher degree of glycogen phosphorylase denaturation. And two, the fact that we didn't see a change from the 6 to 24 hours, that this was all occurring earlier than the 6 hours. Again, we wanted to look at this relationship to the water holding capacity. Again, we just ran simple correlations just to look at these relationships. And the first thing you'll notice is everything's significant. And basically what we found from this, if we're measuring glycogen phosphorylase based off our SDS page work, whether we're looking at the myofibrillar fraction, whether we looked at it in the sarcoplasmic fraction, we found a significant relationship and a moderate to strong relationship with any of our measures of water holding capacity. And we found it didn't really matter if it was at 6 hours or 24 hours. So what did we conclude from this? Well, first of all, overall, that our aging treatment didn't really matter. The relationship between the water holding capacity and our measures of protein solubility or protein denaturation were the same at 6 or 24 hours. And that overall, protein solubility measurements were not related to low water holding capacity in these breast fillets. This suggested that myosin denaturation and breast fillets are not really a distinguishing factor with a low water holding capacity. And then this data also suggested that the increased glycogen phosphorylase denaturation that we saw and the precipitation onto the myofilaments may impair water holding capacity in broiler breast meat. And so what we're seeing in a kind of our take-home message from this was, even though protein denaturation might not be the deciding factor in broiler breast meat like it is in pork or turkey and things like that with the low water holding capacity, if we look at the glycogen phosphorylase and this denaturation, it might have a mechanistic, a coating of the myofilaments, if you will, to impact water holding capacity in vivo situations. And again, we just use the glycogen phosphorylase as kind of an indicator protein. There are other sarcoplasmic proteins that this could apply to as well. So we thank you again for your time.